Alright, let's do lesson 6, spectrophotometry. Spectrophotometry is a very important analytical technique used in both chemistry, biology, and physics. There are lots of applications and importance to spectrophotometry. In this class, we are only going to look at a few of those important applications. Now, in this specific lecture video, we're only going to look at the basic technique of spectrophotometry and a few applications of spectrophotometry, and we'll look at the instrument that is responsible, or the instrument that is used in order to do spectrophotometry. So we're going to start out with looking at spectrophotometers, which are really the instruments that are used in this technique. So a spectrophotometer is the instrument that uses electromagnetic radiation from selected regions of the electromagnetic spectrum. Now, when we mean this is that we have to tell the spectrophotometer that we only want to use specific wavelengths for our technique or experiment. And I'm going to teach you how we do that later on in this lecture video, how we select for the wavelengths that we want to use. Now, these wavelengths can come from the ultraviolet, the visible, or infrared region, and we use those selected wavelengths to analyze the absorption or transmission of radiation by a sample. For instance, let's talk about chlorophyll. Now, chlorophyll is a molecule used in photosynthesis. Chlorophyll has the ability to absorb only specific regions of the visible spectrum, specifically wavelengths that are red and blue, and transmits green wavelengths of light, which is why we see the green colors of leaves, stems, and so forth of these plants. All right. So the spectrophotometer can be used to measure the absorption of light by chlorophyll. Simple as that. All right. Now, for us in our labs, we are going to selectively use a visible spectrophotometer, which basically selects or scans in the visible region. And so we're going to select wavelengths in the visible region for our spectrophotometers. Now, we typically got to use tungsten or xenon lamps to provide the light source in this region, and we'll talk about how that works in just a moment. Here is a picture of a spectrophotometer. This is the type of spectrophotometer that we are going to use in our own labs, and later on I'll kind of get into some specifics of how we use this particular spectrophotometer. Now, in general, most spectrophotometers work in the same manner. You have to have a light source, and our light source is going to be either a tungsten lamp or a xenon lamp, and that's going to provide the light that we need to do our analytical technique. We're going to shine the light on a monochromator. Now, a monochromator is a special instrument in the photospectrometer that is basically a prism that's going to take the light from the light source and break it into its continuous spectrum or its continuous visible spectrum. And then the monochromator is going to select a specific wavelength that we have determined prior to doing the experiment, and only use that light throughout the experiment. And all the other wavelengths of light is going to just kind of filter out, so we can't use that light. So in this particular instance, we have selected the photospectrometer to use only wavelengths at 585 nanometers, which is really yellow light. And so in this case, only yellow light is going to be used in our experiment. All the other lights, wavelengths of light is filtered out. and so we're only going to use specifically yellow light at 585 nanometers. Alright, and that 585 nanometers of yellow light is going to then hit our sample solution that contains our compound that we're using. Now that compound is going to be placed in a cuvette. I have a picture of a cuvette here. This cuvette is holding a pink solution in it. The cuvette is placed in the photospectrometer in a specific slot, which I'll show you later. And light from our 585 nanometer selected range in the visible spectrum is going to hit that cuvette, and the compound that's in that cuvette is then going to absorb some of that light, and the light that is not absorbed gets transmitted through the cuvette and then is picked up by a detector or a photocell. And that photocell is going to determine or tell us how much light that was absorbed and how much light that was not absorbed, and that's going to be digitally displayed. So again, the photocell or detector detects how much of the 585 nanometers of light that was absorbed by the sample. Alright? Okay, so I'm going to erase a little bit of this so we can look at some specifics here. A little bit more details now. Alright, so we have a picture up here, and light from our selected, what was it, 585 nanometers. That's not good. Oops. So essentially this, okay, this light, 585 nanometer wavelength light, hits our cuvette with our solution. This is where absorption takes place. And then the amount of light that is not absorbed is going to be transmitted right out. Alright? Okay, so here's our spectrometer again. Here is some cuvettes that can be used. And when we open up the spectrometer, there is a place where we put the cuvettes in. It's right there. This spectrometer that we also have can use test tubes besides cuvettes. But we're mainly going to use cuvettes. And so we just put the cuvette in that little slot that you see there, and you'll do that in your lab. Okay, so I want to look at some specific variables here. We have I sub O and I. Okay, I and I sub O are two important variables that our photospectrometer is going to determine what those are, and then is going to get us a value called the percent transmittance. We'll talk about what the percent transmittance is a little later. Now, I essentially stands for intensity. All right. I sub O is the initial intensity of the light that hits our sample solution. And I is the amount of light that is not absorbed. Okay? So again, I sub O is the amount of light that hits the solution, and I is the amount of light intensity that is not absorbed by the solution. Okay? Now the other important variable here is length. Now length is the distance that light has to travel through the cuvette, and that will become an important variable later on that we will apply to equations in our analyzing the data from spectrophotometers. Okay? So I sub O and I. Remember, I is just the intensity of light. Okay, here's some other cuvettes. All right, now we've looked at spectrophotometers. Now let's look at the actual spectrophotometry technique. All right, a spectrophotometry, again, is an analytical procedure that uses electromagnetic radiation to measure the concentration of substances. The success of a spectrophotometric technique requires that the absorption of light by a substance being analyzed must be distinct or different from other chemicals in the solution. What this is saying is this, is if I have a solution, I'm going to have multiple molecules that are in that solution. Those molecules, those different molecules, have the ability to absorb light. But we only want one, or analyze one of those molecules. And so we need to find a specific wavelength of light that only the molecule that we're analyzing absorbs. And the other molecules don't absorb it, okay, or very much of it. Now, the wavelength that only our compound absorbs is called the optimum wavelength. And it is the optimum wavelength that we need to select. It goes back to, if we go back to our, it goes back to our monochromator here. We need to select the wavelength here in our monochromator and determine that optimal wavelength that only our compound in our solution will absorb. All right? So we need to determine that idea. So the optimal wavelength for a photometric analysis of a substance is selected by measuring the visible spectrum of our compound. So we take a pure compound, that's the only thing in it, besides maybe water, and we determine the light that is absorbed by that compound, and then we plot a graph of absorption versus the wavelength. And the optimal wavelength is generally determined by the wavelength of maximum absorption. Right? For example, that you may not completely understand this yet, but let's look at an example of this whole process, and maybe it'll make more sense. So let's look at a specific compound called blue 1, or FD&C blue 1. FD&C blue 1 is really a dye. It's actually one of 7 dyes approved by the FDA, which is the Food and Drug Administration, for food and drugs and cosmetics. Okay, so the blue 1, okay, under white light, FD&C blue 1 is a color, basically is a color of light that is transmitted by the solution, or blue is transmitted by the solution. For instance, sports drinks like Powerade over here. Powerade, this particular Powerade is blue, or has a blue look to it, because blue light is being transmitted. So Powerade, or this particular Powerade that's in our picture here, is made up of blue dye number 1, or FD&C blue 1. And so that molecule in that Powerade transmits blue light to us, so we see it as blue. But FD&C blue 1 absorbs certain wavelengths of light that's complementary to it. In order to understand the colors that are absorbed by this blue light, or this blue 1, we have to look at a color wheel. Alright, now here is blue. Blue light is between 450 to 480 nanometers. Alright, so if blue light is transmitted, then the complementary color is orange, which is right across from it. That tells us that FD&C blue 1 absorbs orange light and transmits blue light. All right? So what if we had another compound that transmits, like, say, yellow light? All right? That means that its complement color is violet, and therefore violet gets absorbed by that compound. All right? And if we had another compound, let's say, transmits green, such as chlorophyll, right? Chlorophyll transmits green. Well, what color does it absorb? Well, it absorbs red light. All right, that's its complement color. So we can use the color wheel to determine what colors are absorbed based on the color that is transmitted to our eyes. All right, so back again, F, D, and C, blue 1, that compound transmits blue light, and so it's going to absorb orange. It also does absorb yellow and red light, but mainly orange. And the maximum wavelength at which blue 1 absorbs is 630 nanometers. 630 nanometers, which is in the orange spectrum of range at which is absorbed by the blue number 1. We can see this with a graph here of looking at absorption versus wavelength. All right? You can see that the maximum absorption of light for blue number 1 is right here at 630 nanometers. All right? So what does that tell us? Well, it tells us that blue number 1 mainly absorbs wavelengths of light at 630 nanometers. So that's going to be basically our selected wavelength that we're going to use in our spectrophotometers. Now, in most of the labs that we do, I'm going to give you this value. I'm going to give you the maximum wavelength that we need to dial in on our photospectrometer. So for the lab that you're going to do, your first lab that you're going to do, you're going to use this very wavelength, 630 nanometers, because we're going to play around with blue dye number 1. All right? So let's look at this picture, and maybe we can kind of summarize this a little bit. So in this curvette, we're going to have blue dye number 1 in that curvette. So blue 1, okay? And we're going to select a wavelength of 630 nanometers. And all the other light is being filtered out. And so only 630 nanometer light will hit the blue dye number 1. And we can measure how much of that 630 nanometer of light is being absorbed, and then how much of it is actually transmitted and not absorbed. All right, and that's essentially what we're going to be doing in this lab, this first lab. So here's our digital readout on the spectrophotometer, and this is for blue dye number 1 essentially. All right. Except we're going to use 630 nanometers, and we're going to dial that in. And this is the dial on our spectrophotometer, and we're just going to dial that in until it reads 630 nanometers, and then we're ready to go. And then we put in our curvette, our blue dye number 1 into the curvette, and put it into the spectrophotometer and determine how much light is being absorbed by our blue dye number 1. All right, now what do we read? Okay, what is the digital readout on the spectrophotometer readout to tell us? Okay, and that's what we're going to kind of look at now. So the wavelength of light absorbed by a substance is characteristic of its molecular structure. The intensity of light absorbed depends on the amount of substance in the solution. So in other words, we're generally looking at the concentration of the solution. In general, the more concentrated a solution is, the more light that is going to be absorbed. So the absorption of light is proportional to the concentration of our solution, or our compound that's in our solution. So the more intense the color will be, and the greater the intensity of light the solution absorbs. So a digital spectrophotometer measures basically both what is called the percent transmittance and the absorption. Okay, the absorption is the light that's being absorbed by the compound, and the percent transmittance, we'll talk about what that means in just a minute. Okay, when light is absorbed, the radiant power P of light beam decreases. Now you can think of power as the same thing as intensity, so we can substitute power and intensity together. All right, so the transmittance is the fraction of incident light of the power over piece of O that passes through the sample. Okay, so kind of let's look at this. We know that I sub O is the intensity, or the initial intensity. We can also look at I sub O as P sub O, the power. This means the same thing. And then the light that's being transmitted and was not absorbed, which is I, is also P, or the power. All right? So transmittance is really the ratio of the power over the initial power before it absorbs. Or we can look at it as the intensity that is not absorbed over the intensity before it's absorbed. And that ratio gives us the transmittance. Okay? So that's what this equation is showing right here, is that transmittance is equal to the P's over P sub O. Now the percent transmittance is what we actually read on the spectrophotometer, and that's important. Okay, this is really important to understand. The percent transmittance is what we read on the digital screen of the spectrophotometer. Oops, let me erase this. Okay. Alright, so here we have a digital readout, and it's reading 5.60. That's the percent transmittance on the digital readout. Okay? So when we erase all of this, and when we kind of summarize it all, okay? So the spectrophotometer is going to do a lot of this work for us. Right? It's going to tell us the percent transmittance, which is percent T. And the percent T, all it is, so the percent T is all it is equal to the transmittance times 100%. And the transmittance, T, remember T, is just equal to the power, or the intensity of the light that is not absorbed, over the power or the intensity of light before it's absorbed. All right? And that's all it's going to do, is give us that ratio. Now, once we know that percent transmittance, we can determine the absorption. Okay? The absorption is equal to the negative log of the transmittance. So if we know what the percent transmittance is from our digital readout, then we can use this equation here to find our transmittance, and then plug it into this equation here to find out how much light was actually absorbed by our compound. Alright? So hopefully that makes sense. All right, so there you see the percent transmittance. All right, now what we're going to do is we're going to finish this lecture video up with looking at Beer's law. Now, Beer's law is really an important law and an important equation that I want you to have memorized. Okay? Essentially, we've already talked a little bit about this. The amount of light that is absorbed by the solution depends on the concentration. The higher the concentration, the more light that is being absorbed. So we know that absorption of light is proportional to the concentration. And that's essentially what Beer's law is stating. That Beer's law is really stating that the absorption of light is proportional to the concentration. And the concentration, which is in Beer's law is given the letter C, is in molarity or moles per liter. So all concentrations are in moles per liter or molarity. And it's also going to determine on the length of the path of the sample cell. So remember we said that length earlier in our lecture video was going to come back to us and it was going to be important. The length is symbol in Beer's law is B. So C is concentration and B is the length of the pathway of light through the cuvette that light must travel. Okay? So the equation of Beer's law, is shown right here. Okay? And so that's what we're going to kind of focus on is the parts of this equation. So A is the absorption. That's the amount of light that the compound is absorbing. Then we have this funny looking symbol called epsilon. Epsilon is known as the molar absorptivity coefficient and it has units of moles minus 1, centimeters minus 1. Okay, those are the units. Now the molar absorptivity coefficient is a constant and only changes on different substances. So each compound or molecule has its own molar absorptivity coefficient. All right? And so that is usually given to you before you do a lab. All right? Okay, so we have A, which is absorption, epsilon, which is the molar absorptivity coefficient, and then we have B and C. And we've already talked about B is the length of the pathway of the cell, which will be given to you, and then C is the concentration in molarity. All right? So if you know what epsilon is, and you know what B is, and you know what A is, then you should be able to find the concentration of the compound in our solution. And that's essentially what we want to do in our first experiment, is use Beer's law to determine the concentration of an unknown solution, knowing the epsilon of that compound, knowing the length or the pathway of light that goes through the cuvette, and knowing the absorption of light that the compound absorbs. All right. So, in general, Beer's law tells us that the absorption is proportional to the concentration. All right. Now, what we're going to do is we're going to show you how to do a Beer's law graph. Okay? And the Beer's law graph is something you're going to do in this first lab. Essentially, what you're going to do is you're going to take basically a sports drink with blue dye number 1 in it. Okay? And you're going to analyze the concentration of blue dye number 1 in our sports drink, like Powerade. All right? But to do that, to determine the unknown concentration of blue dye number 1 in the sports drink, we need to have a Beer's law graph that allows us to determine that unknown concentration. So what we're going to do is have some standard solutions with blue dye number 1 in it. Alright? So we're going to have like 4 or 5 different solutions with blue dye number 1 in it that we know the concentration of. Oops. So simply what we're going to do is we're going to graph. So what basically we're going to take one of the standards, say standard 1, and we're going to determine what the absorption of that standard 1 is at a given concentration. So we know what the concentration of that standard is, and then all we're going to do is determine the absorption of that. And we're going to do that for 3 different concentrations of blue dye number 1. And then we're going to plot them. We're going to make an absorption versus a concentration graph. Okay? Erase this. Alright, so we know basically 4 known concentrations. Those are our standard solutions. We know their concentrations. We measure their absorption using a spectrophotometer. Then we graph their absorptions versus their concentrations, and we should get a straight line. A straight line. Like this. Like so. Okay? Now, what we can do then is use this graph to determine the unknown concentration of our sports drink that has blue dye number 1 in it. So if we know the amount of light that the sports drink absorbs, then we can find it on our plot. Okay, this is the amount of absorption, and we can then determine the amount of concentration that it is. But we can also use Beer's law, because if we know the absorption, the amount of light being absorbed by that solution, then we can use our Beer's law equation. Oops. Our Beer's law equation to determine the concentration of the unknown by just plugging it into this equation by knowing epsilon, B, and then the absorption, which we measure using the spectrophotometer. All right? So this is basically it in a nutshell. And I know this is a lot. This is a lot of information. I want you to really study what we've just gone through in your notes, in this lecture video. If you need to look at it multiple times, do so. We are going to spend more time on this idea or concept of Beer's law in class. I'm going to give you some problems to work out, as well as these labs that we're going to do, so that you have a better understanding of Beer's law and how it works. All right? So this is all for now. This is all I'm going to go through at this point. If you have questions, come please see me before class. All right? So that is it.